Thanks very much. Welcome back. We are joined here on the sofa by our three wonderful speakers who've just given us amazing plenary presentations. Mike Clark from the University of Melbourne, Leah Payen from Hospices de Lyon, and Tom Walsh from the University of Washington. Welcome and thank you for your fantastic talks. So, I mean, where do we start? Amazing. I would love to start with Tom. Thank you for talking about intronic mutations of breast cancer and the amazing work you've been doing, really resolving what's going on with some families that have this higher risk. Why are intronic mutations so important at this point in clinical research in breast cancer?
Yeah, so I mean, the BRCA1 gene just spans 5 kb of coding sequence, but the full locus is 100 kb, so there's an awful lot of space for additional mutations to occur, but we can't really, pinpoint those accurately unless we've got long reads, because the introns are basically full of junk repeats. They're just not aligning with short reads. And then the prediction tools are really helpful to pinpoint which ones we need to follow up with long read cDNA. But as you can see, they're just as damaging as a frame shift or a stop codon, based on the family history we've seen so far.
Well, you've obviously made use of tools that can really get all of the information out of the data. And of course, you've done a wonderful talk. Bioinformatic tools are so important in the work that you've been doing in single cell. A general question about tool development. Obviously, you've been thinking about this with relevance to your own work, but how does the development of tools affect sort of more broadly beyond brain development questions into the broader community?
Well, if you think about single cell in general, it's been used for a huge variety of applications in a huge variety of settings. So I guess we feel that the long-read single cell is the same, in that if you create tools that enable people, then they can apply it to whatever questions or areas they're interested in. It doesn't have to be anything similar to brain development at all.
Right. And so what have you found most interesting that you've learned using these full-length isoform data about brain development? What's been the thing that's most excited you in the research over the last year or so?
Well, I guess there's, a very large number of novel isoforms, and we see this in other projects as well. And so sometimes these are really highly expressed or show really specific patterns of cell type specific expression. So it gives you a lot of targets to go after to try and understand a process better, and whether some of these are maybe absolutely crucial to some developmental processes.
Got it. And so full-length isoforms compared to not full-length isoforms that you may be getting with previous generations of technology, what more has that given you?
Well, when you do your standard sort of short read single cell, usually you're only sequencing either the very 3' or the very 5' of any particular RNA. So it's fine usually for gene counts, but it tells you almost nothing about what isoforms are being expressed. Okay. So if you're interested in anything around the splicing variability and understanding the full-length isoform, then you really do need some sort of long-read approach.
So you're missing information if you haven't got those long reads?
You're missing a lot of information.
Fantastic. Well, we hope to help you with that. So, Leah, you've just given us an amazing presentation about pharmacogenetics, so much in there to think about. Now, the word CYP2D6, it's like a superstar pharmacogenetic at the moment. Everyone's talking about why is it so important in pharmacogenetics?
This gene is very important because when it's the protein, the corresponding protein is involved in the metabolism of 25% of the drugs we are using in our medical care. So when we have a deficiency in terms of metabolism of CYP2D6, we can show some toxicities in patients. And if it works too well, the treatment is not efficacious.
Right.
We have no response. So it's really important to study this gene and to determine the variants inside this gene.
Fantastic. And so what direction are we heading in with long-read sequencing in pharmacogenetics? What are the potential impacts on patient care that we could achieve with this?
The potential is very huge because for some patients it's really important to choose the right drugs in the first line of their treatment. In psychiatry, for example, if the drug is well chosen in the first line, the person can work without any problem. If it's not the case, we have some hospitalization time, and it's difficult for the patient to live with his chronic diseases. You know what I mean?
So you were talking about turnaround time being really important.
Yeah, it's really important to integrate pharmacogenomics analysis in the management of the patient to improve the quality of life of patients.
Right. And so there's been a bit of a talk today about adaptive sampling, which for some people means turnaround time and for some people means efficiency. You were talking about adaptive sampling. What does it mean to you?
I mean,
were you excited by Clive's updates?
I was, yeah. But I mean, I've just been through PCR, pull downs, all sorts of alternative enrichment approaches. To do it on the box without any other experiments is just great to me. I've loved adaptive sampling since I heard about it, and it kind of convinced me to go with nanopore, in fact. So.
Fantastic.
But yeah, to obviously sequence more deeply, more cheaply would be great with the new buffers and stuff.
Brilliant. I had a thought when I was watching both of your presentations that the world of sort of risk assessment and the world of having your genetic data, previously available in your health record, are these two worlds too far apart, or do they converge in the future to everyone having a risk score and everyone having their pharmacogenetic information to hand, or are we too far away from that?
I mean, there are population-based screening approaches out there now. In an ideal world with unlimited resources, I think everyone would get BRCA1 and other clinically important genes sequenced at a certain age. And you could certainly build in pharmacogenetic markers at the same time for that sequencing step.
I think so. The worlds are very close because, for example, for some patients, to combine the two information will clearly improve the quality of life of the patient, because maybe they know what's going on for them, and they will better accept the facts, and maybe we can better manage their treatments and so on.
Yeah.
So it's quite close.
Just before you guys came on, someone told me that there was a talk upstairs. I missed it, but someone said that there was a Q70 human genome presented, a Singaporean of Indian descent, and that the presenter was saying that in the future we should all be doing de novo assembly for humans, because the fact that the reference genomes are so biased towards certain populations means there's a 1% error rate if you're not in the sort of typical reference population. So what year will it be where we have de novo, telomere to telomere, whole genome sequence in our health records? 2040, 2050?
Oh.
What do you think?
Yeah, maybe 2050 in some places in the world.
We'd love to get there earlier. We would really love to. So I'd love to hear, what have the highlights been while you've been here today? What's been your favorite talk so far?
So I really enjoyed Dan Kim's talk yesterday. As an RNA biologist, hearing about extracellular vesicles and not only their functions but how you can use them for liquid biopsies, I thought was really cool and really exciting.
Yeah.
Sorry.
No, off to you.
I love Karika's talk. She's setting up genetic testing for the first time in the Caribbean islands, and she's starting with a great approach, nanopore sequencing. She's going to get all the data in one experiment. Copy number variants, single point mutations, indels, and methylation too.
So two talks on breast cancer, yours and hers, quite different. What do you think is complementary between her talk and yours?
I mean, I guess we were finding mutations in the same genes, BRCA1, BRIP1. I mean, she's only really scratching at the surface of her data at the moment, just looking at coding mutations, but she's going to have a lot more data to analyze soon, which is kind of exciting.
Fantastic. Now, what's been your favourite so far?
The first talk about telomeres was very wonderful, and I didn't know so well this field. It's very great, and I think it's important to know, and
Carol
will change things in the future. I think so.
Carol Greider's talk was fantastic,
Yeah.
and she was talking about her career journey earlier, and talking about the fact that she didn't know she wanted to be a scientist until relatively late on in her career. And it was a really wonderful conversation, because she was basically highlighting that science is really open to all, anyone with a curious mind, and you can go on and you can win a Nobel Prize without any intention of doing that when you're sort of 13. So that was very inspirational. It was a great talk. Fantastic. Well, what should we be looking forward to just in the closing hours of the Innovation Day? Any collaborations you want to form? Any people you want to find by the water cooler?
You know, I looked at Mike's acknowledgement slide and recognized Josie Gleason. We actually reached out to her last year about nanocans, but just for quantifying mutant messages. So I'll definitely follow up with her now that I know who her PI is.
Yeah, Josie's still with us in the lab, and yeah, so she's fantastic. So definitely get back in touch.
Cool.
And this is how collaborations are made, right here on this sofa. This is fantastic. Well, thank you so much for joining us. And we are now going to go back to the main menu. You can click on everything, speak to our teams, get some technical help. Thanks very much for joining us, and see you soon. Bye-bye.